Welcome to the Wednesday afternoon lecture series. I'm Polly Joseph, a Lasker Clinical Research Scholar at the NIAAA section of Sensory Science and Metabolism. Today's lecture is special for at least two reasons. One reason is that this is an NIH Director's Lecture, and there are only 3 NIH Director Lectures each year. And the speakers are approved by the NIH Director among all 30-plus WALS nominations to represent the creme de la creme of WALS season. The other reason is that our speaker, Dr. Leslie Vosshall, can finally join us in person. I nominated Dr. Vosshall perhaps 2 years ago now. She was scheduled to speak during the WALS season, but postponed to have that visit in person. So I'm really, really excited to have her here in person and in the flesh. Dr. Leslie Vosshall, the Robin Chemers Neustein Professor and head of the Laboratory of Neurogenetics and Behavior at the Rockefeller University. And also, as of this past February, she became the Vice President and Chief Scientific Officer at the Howard Hughes Medical Institute. Dr. Vosshall has performed groundbreaking work to establish the Aedes aegypti mosquito as a genetic model of organism for neurobiology, with particular emphasis on how mosquitoes hunt humans and the subsequent design of small molecules to block mosquito biting behavior. Her lab takes a multidisciplinary approach spanning neurobiology, behavior, genetics, and genomics. For example, her lab identified the genes that mediate odor and carbon dioxide perception in insects, including orco, a member of the odorant receptor gene family, which is uniquely expressed in majority of olfactory sensory neurons and highly conserved across insect evolution. This research holds promise for combating the spread of yellow fever, dengue, Zika, and emerging viruses transmitted to humans through the bite of the female mosquito. Note that another broad area of interest of her is the olfactory perception in humans. Through the Rockefeller University smell study, she has participated in a large-scale research of human subjects to combine olfactory psychophysics with genetic analysis in order to understand the mechanisms of olfactory perception in humans. Dr. Vosshall received her undergraduate degree in biochemistry from Columbia University and then her PhD in molecular genetics from Rockefeller University. She conducted her postdoctoral training back at Columbia in the laboratory of future Nobel laureate Richard Axel from 1993 to 1997, and then stayed there for the next 3 years as an associate research scientist. In the year 2000, Dr. Vosshall moved to the Rockefeller University in an assistant professor and rose through the ranks, becoming the professor in 2010 and the director of the Kafli Neural System Institute in 2016. She has earned numerous awards and honors and accolades along the way. She became the HHMI investigator in 2008 and subsequently inducted in both the National Academy of Sciences and the National Academy of Medicine. And in 2020, she received the NAS Prado Research Award. The title of her talk today is Bitten: Why are some people more attractive to mosquitoes than others? So Dr. Vosshall, welcome back to the NIH, and we look forward to hearing some of your research. Thank you.
So thrilling to meet Polly Joseph for the second time in 2 weeks. Really thrilled to be here, and I'd like to thank Francis Collins for all of the years of service, so keeping the NIH at the forefront of what our government supports, and wish him well in his quote-unquote retirement, still actively running a lab. So I'm really excited to be here and honored to give this lecture about mosquito biology. And so at around 2008, I pivoted my research program after decades of working on Drosophila melanogaster flies to working on this animal. And we're interested in this animal because this female, Aedes aegypti, is able, through this act of blood feeding, to infect over 400 million people a year. And so we were fascinated by what drove the female to do this behavior, to try to understand it, and try to figure out by understanding it, how can we intervene in this crazy behavior spreading disease. And so Aedes aegypti is the primary vector of these 4 arboviruses: yellow fever, Zika, dengue, and chikungunya. yellow fever being an ancient virus, dengue emerging, and Zika and chikungunya, a little known fact, first emerged in human populations in around 1950, showing that viruses in wild reservoirs have the possibility at any time to jump into human populations by their ability to be basically taken up into mosquitoes and then deposited into humans. And so if you take together Aedes aegypti and Anopheles gambiae and count up how many people these insects are currently killing a year, you come up to about 500,000 people per year, primarily due to malaria. And so it's an important problem to figure out how are these females doing what they do and how can we stop them. And so an important additional consideration is that these are tropical animals, so they require a mild or non-existent winter, they require water, and they require a large density of humans to be able to execute on their business plan. And as the climate warms, more and more of temperate and northern regions become a really fantastic habitat for mosquitoes, including a couple of years ago, there was a massive dengue outbreak in downtown Tokyo. And there have been also Washington, D.C., occasionally you find sort of local bursts of Aedes aegypti. So they're waiting for you, and they're waiting to bite you and waiting to infect you with these viruses. And so that's the question that captivated us. So I'm going to talk about two different stories, one about taste and one about smell. And so I'm going to plunge right into this question about what does blood taste like to a female mosquito? There's a vast number of species of insects on Earth. Only a really tiny, tiny, tiny fraction of insects actually has evolved the taste of blood. And so this work was carried out by Veronica Jovet, a PhD student in the lab, now a postdoc at the Pfizer CTI, with collaborators from Stanford, Pasteur Institute, and Princeton. And so an important takeaway for those getting the SME credit, the one thing that you learned today is that this is a highly sex-specific behavior. So mosquitoes come in male and female. Only the female has the drive to do this behavior for a couple of reasons. So anatomically, only the female has a syringe on her head that can penetrate skin. So males have a feeding tube, but it's blunt, and so they are unable to penetrate skin. I think more importantly, males don't care. So you see the little picture of the female, she's bracing herself against the skin of one of the people in my lab. This is a photo from Alex Wild. You can check out his beautiful gallery of photos on his website. So Alex Wild came into our laboratory and watched our day-to-day, and so this is our day-to-day. So you can see the female intensely focused on, basically has shoved this the syringe-like stylet into the skin, is looking around for blood. The male has absolutely no interest in human cues, absolutely incapable of puncturing something as taut and tough as human skin. And so why is that? It is because males don't produce eggs. So it's the blood is 100% required for these animals to be reproductively competent. And it's a really good trick. So many insects feed on floral nectar or vegetable material, which doesn't have a lot of calories or protein in it. And so animals like ticks and mosquitoes and sandflies, it's the most brilliant thing you can do, where you find something to bite, in the course of a minute, take in this incredibly protein-rich meal, and then go away and digest it, and then convert that to eggs. So the sexual dimorphism is 100% related to the reproductive reality that if today all humans spent the next 2 years indoors and nobody went outside, we would be able to eliminate all these diseases, right? Because you would basically get rid of the reservoir. You would be able to, they need that blood to reproduce. You would crash the population. So it's a really interesting and imperative behavior for them to carry out. And so in the lab, these creatures drink 10% sucrose. We think out in the wild, they may also be feeding from flowers. And so in the course of their life, males never taste blood. Kind of sad, they have no idea what it tastes like. And the females, whenever they need to produce eggs, enjoy the taste of blood. And we've kept these animals alive in the lab for a terrifyingly long period of time. The Dov Shai did the experiment. How long do they live if we treat them really well? It's months. I used to say 6 weeks, but it's like 3 or 4 months. You can keep them alive if you treat them well and give them sugar. So the female faces this quandary about, is it blood or is it sugar? Because these meals are handled differently for different purposes. And so this is what it looks like when you come to my lab. This is what happens when mosquitoes find a little patch of skin. The rest of, this is Emily Dennis, who's now a group leader at Genelia. She loved being bitten. She did all this important work when she was in the lab. And so this is a little patch of skin of her forearm that has been not covered in latex. It's sped up 6 times incredibly quickly. Within a few seconds, the females leave the side of the cage. Something is happening. Carbon dioxide has gotten them aroused. They find that tiny spot. They use their stylet to puncture the skin, and they begin drinking. And so you can see, okay, 6 times sped up, how enormously full their midgut gets with blood. So again, a brilliant evolutionary gambit. Specialize on humans. There's a lot of us. We don't have fur. We congregate in enormous colonies of people, and we always have water around us, so they need water to be able to lay eggs. And so this is another takeaway from today. Only females bite. Get rid of standing water around your house. I now own a house in Silver Spring. The first thing I did was talk to the neighbors about don't have standing water. So it's the standing water that makes us such incredible, great hosts. And we call this host seeking because we really are hosts. We do everything we can to welcome them into our lives. We will hike without, we'll hike almost naked. We have water around the house for them to breed, and we have delicious blood. And so you know that if you get a blood test, you get sort of a profile of what is in blood. And so what I would remind people is that our blood, even if you don't have a sugar imbalance, has relatively high levels of sugar in it. So blood does have glucose in it. And has all these other constituents that these animals are looking for. So it's the proteins and the lipids that are driving egg development. So you can make an artificial blood meal that has 3 human blood proteins. You add ATP and some sodium bicarbonate and sodium chloride, and you can keep them alive in the lab for generations. So those are the constituents they need: 3 human blood proteins, sodium bicarbonate, salt, and ATP. And so this is sort of the question about, okay, what's in floral nectar? In this beautiful study from 2006, ecologists went to South America and asked, okay, what's in plant nectar? And how does it relate to the different pollinators that feed on it? And so what you notice is that their nectar is extremely rich in sucrose, fructose, and glucose. That's kind of what it is. It's basically what we feed the mosquitoes in the lab, 10% sucrose. And so the females have this experience of all this stuff in blood and this relatively sugary, sort of simple meal of sugar. And so we've been asking these questions about how do they tell the difference, given that sucrose is a common ingredient between them. And so I'll tell you a little bit about the neurobiology of that. So one thing that's really cool and interesting, we have one tongue and we have one stomach, right? So anything that you put in your mouth, if it's coffee or steak or cake, everything is sensed by the tongue, and everything goes into your stomach. Mosquitoes have 2 separate tongues. They have a tongue for blood and a tongue for sugar. And so I already mentioned the stylet is this sharpened syringe that is the only part of the mosquito that goes under the skin. And so it's covered with sensory neurons that detect blood. The labium is this soft tongue that will sit, and then when it's in a flower or in the lab, we'll taste the sucrose. And so what's even crazier is that here we did the experiment where we fed them first green food dye sugar, and they taste that with the labium. And you see that all of that sugar goes into the crop, and then we give them a little kind of chaser of blood, and all the blood goes to the midgut. And so it's this remarkable thing where they have 2 stomachs. The midgut contains the blood, which is then digested to trigger egg development, and the sugar is kept in the crop as just like a metabolic, like one of those gel packs if you're a marathoner. So I have so many questions here about, first of all, how do they tell the difference? How do they shunt it to stomach 1 or stomach 2? What is the signal there? And the Dovshai in the lab is actually looking like some sort of internal sensing to figure out where it goes. So a key point, again, the crop is small. The midgut can stretch enormously, like enormously. So you can see here that unfed animals will, if you basically look at how much they consume, these animals consume 3 microliters, which essentially doubles their body weight. And if you offer them sugar, they sip it. Again, probably constrained by the size of the stomach that's receiving it. I'm going to do a little sidebar here. This is this work of Rachel Galoon, who I think just turned 98 years old at the University of Jerusalem, Hebrew University of Jerusalem. And she made this discovery that ATP is one of the most exciting tastes that an insect ever encounters. So there is ATP in blood, and it is a key ingredient in our artificial blood because it triggers feeding. They just love the taste of it. So much so that we can make this mixture of ATP, sodium bicarbonate, and sodium chloride. And here's a picture of a mosquito that's drinking it. So she'll puncture this membrane parafilm as if it's skin, and she's filling up on this mixture. So we know that this taste system can be activated by ATP and some buffered salts. And you can see here when we look at the percent of females that get excited by this, you can see that they're making decisions. So if you give them high concentrations of sucrose, they do not engorge on it, does not go to the midgut. If you give them saline, they don't engorge on it. If you give them blood, most of them do, and you give them ATP and saline, they love it. So the sensory system is handling these two very different substances in the same way and is rejecting sucrose. So now let's turn to the syringe. So this is a film from Valérie Chaumet at the Pasteur Institute. This is a malaria mosquito stylet rooting around under the skin of a mouse looking for blood, kind of like the phlebotomy suite down the hall at the clinical center, although she's really good at it. And she's not, she's above the skin. She's bracing herself on the skin. There's this organ that has lots of mechanosensory and chemosensory neurons, is able to do this, to be able to find that capillary. So truly, truly remarkable. And so Veronica set out to figure out, what is this thing tasting? And how does it avoid getting confused and sucking the substance and putting it in the midgut? Because if you put sugar in the midgut, then you interrupt reproduction. So the first thing that we did was teamed up with Lindy McBride and Jile Zhao at Princeton. They made this beautiful strain that allows us to label all neurons with a red fluorescent protein and GCAMP, the genetically encoded calcium sensor. So we could ask how many neurons are there and how do they respond to different tastes. And you can see at the top is the female stylet, has about 25 neurons. You can see the scale bar, it's tiny. It's a tiny, tiny structure, a single layer of neurons, transparent. It's like a dream for neuroscience. And the male has fewer than 10 neurons. We think none are chemosensory. So the male, it has probably some flow neurons to see as it's sensing the sugar that it's drinking, but the female really is set up to taste blood. And so Veronica set up this imaging protocol where she pulled the stylet off the head of the female mosquito, put it in a bath, and used this microfluidic device called a biopen to shoot sheep blood at the stylet to see, are there 3 neurons in the stylet, or half the neurons, or all the neurons? What does it look like when the stylet tastes blood? And so in this video, you have the tip of the stylet at the right, and in a second, you'll have just a little bubble of blood. And you can see the baseline fluorescence of GCAMP is elevated in approximately half of the cells, and it's highly reproducible. So about half the cells get really excited by the taste of blood. We have no idea what the other half do. It's kind of interesting. Have no idea what they do. Maybe they're involved in bad things that they would taste that they would not drink. So I mentioned a couple minutes ago that females can't tell the difference between blood and this completely protein-free meal. They love it. They'll engorge to the same level. And so we ask, do they love it to the same level because it activates maybe the same set of neurons? Maybe that's the message. And that's what Veronica found. And so again, in this film, you'll see in a second blood being applied to the stylet. There you see the happy neurons lighting up. And then in a second, you'll have this protein-free meal containing ATP. So if anything, they really like the taste of that ATP. If anything, it activates the cells more reliably. And so we then decomposed this mixture into ATP, sodium bicarbonate, sodium chloride, and found, again, you can see that half of the cells in the stylet respond to none of these substances. And you can look at the cells that respond to blood and start breaking them into different clusters. So we have a whole set of cells that respond only to ATP, which is fascinating. Insects do not have purinergic receptors. So we sense ATP with P2X and P2Y. Insects do not have these in the genome. So this is a fascinating thing. We do not know how these neurons are sensing ATP, but clearly they have some sort of an ATP sensor. Then there are cells that only respond to sodium bicarbonate, also an interesting chemical substance. Neurons that respond only to sodium chloride. And then these fascinating integrator cells, which will only respond to glucose in the presence of these two other solutes, sodium bicarbonate and sodium chloride. And so Veronica began making gene-targeted mosquitoes to try to figure out, can we find markers for these cells? And so she was able to make a line where all neurons expressing this chemosensory receptor called IR7A express markers. You can see the very cute little cells there. And all of the cells expressing IR7A are sodium bicarbonate cells, which is interesting because then we could be on the hunt for what is a sensory receptor for sodium bicarbonate. She then made another line. Here you see the 4 cells expressing the chemosensory receptor IR7F. These are really interesting neurons because, again, they only respond when you have glucose and sodium bicarbonate and sodium chloride present. So they don't respond to glucose alone. They only respond to the mixture, suggesting that there must be multiple chemosensory receptors that are kind of listening in, which is sort of what it looks like here. So these are cells, again, no response to glucose at all. So 4.5 millimolar glucose is sort of blood glucose. You only get excitement when you have all of them. And we thought this was a key principle that if you don't want to get confused between blood and sugar, make sure that the stylet does not drink pure sugar, as would happen with nectar. And so we think that this is really the key, that the integrator cells really integrate information. And so if you do not have the blood components, the cells do not get excited. And I think a really striking demonstration of this, now we put 300 millimolar sucrose or fructose or glucose. Each of these rows is a single female. Each square is a single neuron in the stylet. At the bottom is potassium chloride, which sort of non-specifically activates all the neurons. And you can see with a few exceptions, primarily with glucose, we don't find a single neuron that responds to these high concentrations of sugar. So I think that the key to this problem is that you have neurons in the stylet that are really choosy, that will completely ignore the taste of high concentrations of sugar unless you also have these blood components. And so we're super excited about what are the mechanisms within those neurons where they don't care about 300 millimolar glucose, but then you add a little bit of sodium chloride or sodium bicarbonate, and all of a sudden they'll fire, and you'll cause this feeding. And then also Nadav Shai's project about how on earth do you take like the steak on one part and then the cake in the other part. It's really cool. Okay, so now I'm going to turn to, last week was a week, so we had this paper come out that was pre-printed in January. So when this paper was on bioRxiv, we did a little tweet storm about it. We had like 20,000 people go to the page, mostly members of the general public, and like thousands of people downloading the PDF, which is unusual. So we knew when it ultimately finally wound its way through the endless grind of peer review that when it came out, it would be a thing. And it was, so we were like on USA Today, did a print version of it. The NBC Nightly News crew was in the lab. And I knew that this would be a big story because this is the question that 100% of the people in my life, strangers on a train, which was a really good movie, and scientists, anybody wants to know, why am I always bitten? When I'm at a picnic, why am I always bitten? It seems to run in my family where half the people are never bitten and the other half are always bitten. So what is it? So I have to turn back to what makes us attractive to mosquitoes in the first place. And so an important thing is that we cannot help, but we're helpless. And I want to say that from now on, my talk is a message of hopelessness and despair because there is nothing that we can do about any of this. So carbon dioxide is the most potent elicitor arousal signal for the mosquito brain. Tiny, tiny amounts, tiny bits and pieces of carbon dioxide, as Trevor Sorrells, who is now a professor at Yale Genetics, showed, like a few seconds of CO2 will flip them into the state of arousal for 15 minutes. So it doesn't even matter if you run away. Like they're still excited and will keep hunting you. Other bad news is that we produce a lot of body odor. So our skin is covered by this natural moisturizing sebum, which contains waxy substances and a lot of lipids. So you don't even need to buy all that stuff that you put on your skin because you're producing it every day. And you have mostly beneficial bacteria on your skin whose job it is, first of all, to keep your skin healthy, and the way that we reward them is with sebum. So the bacteria digest the sebum and then produce the body odor. And then lastly, we happen to be also warm-blooded, and mosquitoes love warm blood. So if we put out dishes of cold blood, nobody wants it. So they like the warmth, they like the CO2, they like the body odor, and also visual contrast. So people ask me, what should I do? When should I hike? It's like at the height of the day when it's really hot, and cover up. And it doesn't matter if you can wear perfume, nobody cares. The mosquitoes don't care. You can't, don't hike, stay indoors. So is it true that some people are more attractive to mosquitoes than others, or is this observation that everybody has really something that isn't a thing? And so, in fact, there are many studies that show, either in laboratory settings or in the field, that some people are much more attractive to mosquitoes than others. So here's a 1993 paper. I do not have a PMID here, so I made a mistake. My new thing is no journal names on talk slides. No journal names on talk slides, but so I already broke my rule. This paper, what they did is they had volunteers sleep in the field overnight in little structures, and then they counted how many mosquitoes were attracted into a trap in the hut. And they sort of randomized who was sleeping in which hut, and found that these really striking differences. And so if you look at person K on the right, had a really great night's sleep, because very few mosquitoes were attracted into person K's tent, compared to person A, who attracted almost 50 mosquitoes. And so these are, this is like a five-fold difference in, these are sort of native mosquitoes flying around. They're hungry. They smell the carbon dioxide and the body odor. They go in, and there's some sort of interesting choosiness. Like person K has blood, person K is alive, but there's something that makes person A much more alluring. And so we set out to figure that out. We being Allendale Baldia, this was a 7-year study. I think a lot of people feel like, whatever, just test it, figure it out. But it took her 7 years. Takeshi Morita made all of the mosquito mutants. Another message of despair and hopelessness that basically you cannot do anything to mosquitoes to get them to not find us. So that's his data. And then Laura Dedman did over 2,000 trials asking, do mosquitoes prefer person 1 over person 2? With bioinformatics support from Caroline Zhang at our CTSA. I'd like to thank the NIH for funding our CTSA. And then the Memorial Sloan Kettering Metabolomics Core that did all the chemistry. And so how do you ask who is more attractive to mosquitoes than others? You could invite people into our tropical room for 2,500 visits over the course of 3 years and have them spend 8 hours a day in a room that has 80% humidity, 28 degrees Celsius. But as it turns out, nobody wants to do that. It's horrible. When the NBC Nightly News crew was here, they were complaining the entire time about, like, our lenses are fogging up. It's horrible. We're sweating. And so to actually do this study, we needed to find a way to get the humans, to have them donate their scent to us, because we knew that it was going to be scent, because carbon dioxide and heat are generic. Every animal that's alive produces those, every warm-blooded animal. And so it's a well-known secret in the field of the chemical senses that nylon is like the premier material to collect human body odor. So it's also cheap and available commercially. And so we were, every week, like pallets of no-nonsense knee-highs coming into the lab. We cut the toes off, and we have people wear the stockings for 6 hours. And again, it doesn't really make any difference what you do to cover up the scent. Whatever is soaking into these stockings is enough to allure mosquitoes. And so this took 2 years for Ellen and her team to work out. It's basically, how do you ask if person 1 or person 2 is more attractive? So the setup is that you take 50 females who have never had a blood meal. So they're raring to go. They're extremely eager to find a human. You release them into this box, and then they can choose door number 1 or door number 2, and the only information they have is the smell coming from the stockings. That's all the information they have. And so I'll show this little video. And I really hope that the sound is not playing out in the world because it's a TikTok thing from our lab. So this is Emily Zeladon, day after day after day, chopping up these pantyhose into little pieces. And that is all that the mosquitoes need to be able to tell the difference between person 1, person 2. And then this is taken in our tropical room. There's Laura Dedman stuffing in the cues. It's 2 assays running in parallel. So you have like person 1, person 2, person 3, person 4. She opens the gates like an aqueduct racing track. The mosquitoes come out. They immediately fly toward those 2 doors. You can see them checking things out, and then they immediately go into the trap. And so Laura did over 2,300 trials like this over 3.1 years. She's amazing. And so some people really are more attractive than others. This is a simple version of this experiment. We put person 33's stocking into one port and a completely unworn stocking in the other. And we ask, can the mosquitoes tell the difference? And you can see that subject 33, who's the star of this whole story, reliably attracted enormous numbers of mosquitoes. If you look at person 28, they don't smell any difference from a completely unwrapped stocking that you just got from the pharmacy. Like, there is nothing on that stocking, so the mosquitoes cannot tell the difference. So it's really remarkable that you go from basically nothing to somebody who's incredibly attractive. But there's some sports fans in the lab, so we thought, like, let's do a March Madness bracket tournament where we compete every person against every other person. And so this took, and multiple trials, so these are all the data. At the left, again, we're looking at the percent attracted. So at the very left, you have subject 25 versus subject 25. Very happily, the mosquitoes can't tell the difference, so we don't have a side bias. You can see all of these people at the left where there's not any significant difference between them. And at the right, there are all these significant preferences. So, and if you look at subject 33 in that maroon color, they win every game. They win every game, and then we can sort of apportion people into these different categories. And so if we come up with this attractiveness score of like how many of these games did subject 33 win, they are almost 100, they're over 144 times as attractive than the least attractive person, which is person 28. And so I think an important thing, as you look at these numbers are scaled according to their attractiveness score. So what we found is that people have a very poor sense of how attractive they are. I would have people say, like, oh, I'm a mosquito magnet, and then we'd come in, and they were really like person 25, like unimpressive. I, too, thought, like, I am the queen of attractiveness. I'm somewhere at the low to middle range. And the idea is that it's all relative. So if you are subject 31, and you're hanging out with subject 19, you'll always be bitten. But if you're subject 33, you're hanging out with subject 33, and you're 31, you'll never be bitten. And so this is why, as far as human subjects recruiting, self-reporting was very unfortunately completely useless. So if you really need to know if you're a magnet, you have to send us nylon stockings. So, but actually, please don't send me stockings. I have never gotten so many emails from the general public as in the last week, because everybody has their own ideas about what it is, which is hilarious. I'm like, as I'll show you in a minute, it's the level of carboxylic acids on the skin, but people are like, no, I have my own ideas. So. And what's remarkable is that subject 33 and subject 28 were this property of being incredibly magnetic to mosquitoes was extremely consistent. So over the entire course of the study, there was never a game that subject 33 lost. So whatever this is, it's a highly stable trait. And I want to note that James Logan at the London School of Tropical Medicine and Hygiene did a twin study to show that there's a high heritable component to this phenomenon, that identical twins will have very, very highly correlated mosquito magnetism scores compared to fraternal twins. And so the question is, how are mosquitoes making these judgments? And so I'm going to turn to sort of the core of what the lab does, which is the chemosensory receptors that the mosquitoes have in their nose equivalent. And so over the course of really, from my postdoc days at Columbia from the late 90s, we know that insects use a totally different mechanism to sense chemicals. So they use ion channels rather than G protein coupled receptors. There's 2 massive superfamilies of odor-gated ion channels, the odorant receptors and the ionotropic receptors. The ionotropic receptors are homologs of glutamate receptors, NMDA, kainate, and AMPA receptors that have been repurposed for smell. And in both these cases, these ion channels form heteromultimers that have a common subunit and a variable subunit. And very conveniently, you can delete ORCO, which is the co-receptor, and destroy the function of all of the hundreds of odorant receptor genes, because without that co-receptor, none of them can assemble into ion channels. And it's the same story for the ionotropic receptors, that you need a co-receptor and then the ligand selective subunits. And then interestingly, they sense two totally different classes of compounds. So the OR sense these things that you have in the perfume in your closet, and then the IR sense the foot odor that you have on your socks and the smelly cheese in your fridge. So they're acids and amine receptors, which makes sense because they're converted from glutamate receptors. So we tried to break the mosquito. So we first knocked out ORCO, and we know from work from almost 20 years ago that these mosquitoes still are really attracted to humans. And so we asked the question, can these mosquitoes that have lost half of their nose, can they still tell the difference? And the answer is yes. So I defy you to tell the difference between the top and the bottom data sets. So they're equal order of magnitude of attraction and perfectly able to tell the difference. And so that put our attention onto these foot odor and cheese odor receptors, the IRs. And so Matt DeGennaro, when he was a postdoc in the lab, knocked out this co-receptor called IR8A. He did it in his own lab, So in Matt's lab, he made IR8A mutants. And we asked, can the 8A mutants still tell the magnet from the non-magnet? And the answer is they absolutely can. So they're highly reliably able to tell the difference between 33, 19, and 28. You will notice that they're impaired. So not being able to smell this class of molecules does impair them. And then Takeshi Morita made these two additional mutants, knocking out these ionotropic receptor co-receptors, and it's the same story. So again, if you look at at how subject 33 is perceived, there's an overall decrease in the level of attraction broadly if you knock out any of the IR co-receptors, but the animals are still able to find people without any problems. And so what we figured out is that there's still intact discrimination between humans, although IR seem to contribute to overall attraction. But how can this be that you can take away half of the olfactory system and then another half and another half, and they're still able to smell us? And so I'll do a little side journey into something that we reported earlier this year, work of Margot Hurry, Olivia Goldman, and Meg Younger, which is that the reason that the mosquitoes are very difficult to break is that they do not follow this rule. So this is a foundational principle in the sense of smell, that in the nose, and so honored to have some chemosensory people in the audience. So in the nose, every smell neuron expresses one and only one receptor that determines what that neuron smells. All the neurons expressing that receptor converge to one place in the brain that then encodes the quality of the odor. And so this has been the dogma. And so Meg Younger wanted to play with the system in the mosquito and completely innocently started making gene-targeted lines to be able to say, like, I want to label like the red neuron separate from the blue neuron, starting with the OR co-receptor and the IR co-receptor. Everything we knew in the fly would say they would label two different populations. But oh no, not true. So essentially, these are pictures of the brain of a mosquito that's been gene-targeted. All of the neurons expressing IR25A at the bottom are labeled in green. And you can see that the vast majority of glomeruli in the brain are innervated by IR25A neurons, and the vast majority of those also are targeted by ORCO. And so you have this code where you have all the glomeruli seem to be getting input from ORCO and IR25A, which could be co-convergence of neurons expressing like the IR versus the OR, or could be that they're in the same cell, so breaking the buck and axle dogma that I grew up with. So to ask that question, Olivia Goldman carried out single nucleus RNA-seq and showed that mosquitoes break the buck and axle dogma. So each row is a specific gene, and so this is sorted, all of the cells across the top of the cell, so all the cells in this particular experiment express ORCO. You could look at cluster 38, they also express IR25A and IR76B, OR82, IR41L, IR41M, OR81, OR80. Like this would make, if you were a mouse smell person, this would make your mind explode because this should not happen, because everything in the mouse goes through extraordinary efforts to prevent this. And we see this again and again and again, that neurons express multiple receptors. And Margot Hurry carried out additional experiment to just actually look, is it true? Like we saw it with gene targeting, we saw it with single nucleus RNA-seq, and she sees it by in situ hybridization. So she can find neurons at the top here that express OR8, IR76B, and ORCO, and in the middle. And so again, I think the take home here is that mosquitoes have taken a different path. So many of the neurons express not only multiple ligand selective receptors, but both the OR and the IR, so the perfume and the stinky cheese in the same neuron. And we think that this is why it is impossible to break them. And I should also say that Chris Potter's lab, at the same time as we made this discovery in the mosquito, showed that actually it's much more complicated than originally thought in the fly. So now I'm going to get to the reason why we were on USA Today, which is what is different about the magnets. And so, as I said before, our skin is covered with sebum, and there are skin bacteria that are responsible for producing body odor. And we think that there's something in the body odor and the skin that is causing these differences. And if you look at the kind of odors that we give off hundreds of different odors. And we focused our study on the things that would be detected by IRs, which is the acids. And so teaming up with Justin Cross and Dan Baumler at MSK, carried out basically chemistry to figure out what's on those stockings. The stockings, what's on the stockings? What's different? And the answer was like immediately apparent. So you look at subject 28 was not discriminable from an unworn stocking by the mosquitoes or by me. Like the chemistry is the same. There's nothing on the stocking. Whereas subject 31 and 33 have a lot of substances on the stocking. So it's like from day one, there's just a lot more stuff on the stockings. And so what are these substances? And so when Ellen applied this filter that you had to be enriched in all 4 experiments, she came up with 40 molecules that were highly enriched in the high attractors. And all of them were these long-chain carboxylic acids, which when metabolized by bacteria produce big smells. And so these are just some of the raw curves from the GC mass spec chromatogram, and you can see just the huge quantities of these 10 to 21 chain carbon carboxylic acids that are highly increased in these subjects. And if we show this as a heat map, where red means high abundance and blue means low abundance, you can see in that little quadrant from a 10 carbon to a 20 carbon that the highly attractive subjects 31 and 33 have massive quantities of these carboxylic acids. And in general, the low attractors have little or no of these. But we wanted to figure out, can we show that it's true in a validation cohort? For years, we've been working with these poor 8 people in the lab who every day had to wear stockings. So we broadened the search to new volunteers who came external from the lab. We recruited 54 of those people, and then basically asked how attractive they are. It's extremely easy to find mosquito magnets and then the low attractors. And so then Ellen repeated this experiment to ask what is on the stocking. And so again, she found that you get most of the action in the enrichment is in the high attractors, suggesting that they're just making more of something. And again, in this case, the answer is long-chain carboxylic acids. And so here's what it looks like for some of the subsets of people that we displayed in this paper. So again, heat map red means high levels of these substances, and heat map blue means low levels. And so you see that there's a super strong correlation between having a lot of the carboxylic acids and being a mosquito magnet. Those of you who are actually looking at this slide right now will see this. And so subject 90 is an interesting biological curiosity, because subject 90 is producing massive amounts of 10 carbon to 20 carbon carboxylic acids, but is extremely unattractive to mosquitoes. And so I can't give you the answer, but it does show that there's clearly, we've shown a correlation here. It's clearly not completely causative. Maybe subject 90 is giving off, maybe something else about their skin changes how those carboxylic acids are produced into volatiles, or maybe they are making the next product that we all need. Maybe they're making the best repellent that ever exists on Earth that's able to overcome this. So it's a really interesting idea. And so in the last experiment, what Ellen and her team did was ask if it's really the amount of the substances on the stocking, we should be able to cut very high-tech, pair of scissors, nylons, cut it into smaller and smaller pieces and ask at what point do you equalize attraction. And we could also kind of get at this idea about, well, maybe subject 28 is producing a repellent. So we could kind of get at this idea about, is there an actual repellent effect? And so the answer is that it really is the amount of the substances, because you can cut a tiny 1/32 swatch, tiny little piece, and only at that point do the mosquitoes not have a preference. And so this just came out last week, and again, the general public is extremely interested in this. I have been like a little busy because I have this other full-time job in addition to running my lab, and I now have people emailing me like, hello, I'm waiting for a response to my question about, so the public is super interested in this topic. And so the information I can give them is that, sort of in this cartoon, that having high levels of carboxylic acids on your skin changes something about how attractive you are, although subject 90 breaks that rule. And this is meant to indicate that this woman who is like hiking in the spring, or playing beach volleyball in the summer, or leaf peeping in the fall, will always be the mosquito magnet, and that this phenomenon is highly stable. And so I'm always asked, okay, so what do we do about it? How can we translate these discoveries into something that's useful? So I think subject 90 is an interesting subject for chemistry. And then also, if you can alter the skin chemistry to make us more like subject 28 or subject 19, interfering either with the amounts of carboxylic acids or the bacteria that produce smells. And so with that, I'd like to thank my amazing lab that has stood by me as I took this big job at HHMI, and we're still holding it together, doing great science. And thank you for your attention, and NIH for the invitation. Thank you.
Is there a breakdown in the mosquito blood source by age, sex, and diet?
Of the person.
Yeah.
I honestly do not. I'm going to try to like free associate a question. So, so we don't see any effect of the gender of the person who's being bitten. So that's definitely not a factor. We didn't control for diet, and so subject 33, regardless of what they ate, was highly attractive. So I apologize. If the person was here, I could ask them to clarify, but that's all I can do.
We'll take one question in the audience.
Yes, very good. What does the mosquito get out of this discrimination power? Is one human actually more nutritional than the other, or is it just more likely to be human?
Yeah, it's a great question. And I think in the field, there's like, one version is that it's an epiphenomenon, and they're not evaluating the quality of the meal. Like, my slight bias is like, of course, they're evaluating something. So I think it would be cool if there is something in the blood, maybe that relates to the first question, that maybe the carboxylic acid is signaling something that represents some quality of the meal that over the course of many generations would confer a benefit. It's really hard to do that experiment because I would need to get subject 33 to be in my lab or give blood for 10 years and then figure out at the end of it, like, is that part of the colony, like, have higher fecundity and longer lifespan? But my bias is like things like this in nature don't happen by accident. That for sure those people they're much more vulnerable to be infected by Zika, dengue, chikungunya, and yellow fever. For sure, like, it's a huge liability for them. But yeah.
Thank you. There's another question here. Can the stylet detect the direction of blood flow? Does it matter if the stylet lines up with or against the blood flow?
That's great. So we know that there are two chemosensory. We know that there's 40 neurons. We know that 2 of them are mechanosensory. So we do think that the stylet is able to sense flow. In videos I didn't show you from Valerie Shumet, the Anopheles mosquito has 2 strategies for getting blood. One is what I showed you, which is like the phlebotomy. And the other is the machete. So they just take the stylet, literally like a machete, slash and create a hematoma, and then drink the blood. So at least in Anopheles, it seems like they're sensing the blood. There's experiments that Adriana Rosas in my lab has done where she has been replicating those movies and has found in preliminary work that if the mouse, when the mouse is alive and there's the heart is beating, the female is able to find the stylet. But if, regrettably, the mouse dies in the course of the experiment, then the animal is not able to sense the flow. So we think that there is something about the blood flow that is stimulating the attraction.
We'll take one question. Oh, there's, yep.
Thank you, Dr. Vosshall. Hi, I'm Ben Sumner. Sorry, just thinking about the blood feeding. But with the smell, first off, would your derivatization and GC procedure, would it have picked up smaller things like KGA, ketoglutaric acid or something like that?
So this is the decision we had to make. So the answer is no. I'm not a chemist, but in consulting with Justin Cross, you have to make some decisions up front. As you do in protein chemistry, you're looking for like big things or small things. And so the derivatization was really specifically targeted at this class of acids, of long chain hydrocarbon. Many of my colleagues in the field have focused on molecules like the ones that you mentioned. It uses totally different chemistry, totally different GC columns. And so we don't know. We haven't rerun all of our samples to sample that other part of chemical space.
Thank you.
But it's a great question. I know where you were going with the question, and I can't answer it.
Thank you.
So I was really interested in your data that mosquitoes feeding on ATP feed just as well as blood. So do you think that is the active ingredient, or are there differences between real blood versus ATP feeding in terms of fecundity or?
Yeah, it is really. It's really striking. They love ATP so much that you can't. When you're watching them, they don't know that they aren't drinking blood. However, because buffered ATP has no protein in it, right, they'll never produce eggs. So this would be another way to, if you wanted to collapse mosquito populations on Earth, just give them buffered ATP because they won't produce eggs. So fecundity effect is no fecundity. I don't know why that is. Again, Adriana Rosas has been asking, as the stylet is slashing through the tissue, is the tissue releasing ATP, and has found that it does. And so ATP is part of the tissue damage signal. So we think that that's why ATP is important, because it's it gets their attention. They're not just slashing through pudding, or that there's something about the ATP that stimulates them to drink. That's kind of how we're thinking about it.
Cool, thanks.
We're getting quite a number of questions about the microbiome.
Okay, good. None of which I can answer, but I'll try.
Yeah, so just to combine them, so do you have a comparative microbiome data for number 33 versus number 28? And can they tell us something about the origins of the differential carboxylic acid species?
It's a great experiment, and for years I've spoken with Julie Segre, prominent researcher here, microbiome, about doing that experiment. We just haven't done it. We know from all of the people working on the human skin microbiome that everybody has a unique garden of bacteria, and it almost certainly contributes to these differences. People have done it. We have colleagues in Europe who've done this experiment. And the answer is that you do find differences. So highly attractive people and weakly attractive people indeed have differences. And the skin bacteria, when you incubate them in sweat, produce different smells. So almost certainly we would find differences, and we haven't done it.
Thank you. Another interesting question, do repellents like DEET change attractivity in these assays? So if we're using those repellents. So DEET is a surprisingly controversial word. If anybody says DEET at a smell and taste conference, people just start fighting in the aisles. So there's no consensus agreement about how DEET works. So some people think that it's a repellent, so it smells bad to insects. Our data suggests that it interferes with the ability of that protein I showed, Orco and the ORs, to actually encode odors. And there's a new cryo-EM structure from Josefina Del Marmo and Vanessa Rutter showing that DEET goes into the odorant receptor and changes its conformation, presumably. So what happens when you put DEET into this assay is that the females will avoid it.
So they won't go into door number 2, unless they're orco-mutants, in which case they'll just happily, if we did it with the orco-mutants, it wouldn't make any difference if we had DEET there or not.
One more interesting question. When you compare carboxylic acid level between subject 31 and 33, subject 31 has higher levels but not as attractive. Does this mean that there's something else playing?
Excellent, yeah, and a person who's very attentive. So I highlighted subject 90, who's a real outlier. Subject 31 is also an outlier. So they were, when we started getting the raw data, we did everything de-identified. So we didn't know whose chemistry was which. All of the nylon stockings were de-identified. So when we first saw the GC mass spec data, I was sure that the chemistry of 31 was 33. So. But for sure, 31 has the highest levels, still highly attractive, but not as attractive as 33. So I think clearly there are other components. You see this with subject 90. Mm-hmm. So I think that there are almost certainly, again, maybe subject 31 makes a little bit more of a repellent that kind of counterbalances.
Does adding a stocking from a low attractor, especially subject 90, to one from a high attractor decrease attractiveness?
That's an excellent experiment, and so the hypothesis there is that subject 19 is producing a repellent, and so then you could counterbalance. Ellen, it's not in this paper, did a little bit of this, and we didn't find any evidence that adding 19 would mask or decrease. So we really do think it's a positive signal. We didn't do that with subject 90. Which a couple people have emailed me. I get emails also from, it's great that people care about this work. So I've gotten a couple suggestions from people to do this experiment with subject 90, if you mix 90 with 33,
So another question is, do we have any up here? Oh, okay, we have one here.
Hi, I wanted to know if you had any documented cases of someone changing from a high attractor to a low attractor or vice versa. Or would you expect it?
So in our study, we did not. So every single person maintained their place in the hierarchy of where they were in the round robin. But again, turning to people's experience out in the world that could be clinically tested, I have this huge collection of members of the general public writing to me, including with this recent burst in the last 2 weeks. And so pregnancy is a factor. So during pregnancy, the rules change. So, and pregnancy is a good longitudinal thing because it begins and then it ends. And so there's actually good published data that your attractiveness changes during pregnancy. That's not done by us. Beer is another one. So if you ingest beer versus water, your attractiveness goes way up. And so you can do these things with diet. B vitamins is like this other thing that is not true. So everybody tells me you just eat a lot of B vitamins and you won't be bitten. So there's actually no good evidence for that. So I guess just don't drink beer because there's good data that drinking beer increases your attractiveness.
So and there's 2 questions. One is, does blood type, an individual's blood type, make a difference on the attractiveness?
Yeah, so I would say the top 5 hypotheses that are out there are B vitamins, garlic, bananas, being female, and blood type. And there's no evidence for any of those. So there's, with blood type is really difficult because if you think about how to recruit into a blood type study, how many people you would need to recruit to get the power to cover all of those. We did a study that's still not published with 100 people to try to attack the blood type problem, but you don't, for the rare blood types, you have to specifically recruit for the very ultra rare blood types to be able to say anything. And so there's a couple papers, one that says it's blood type and the other one says it's not blood type, and they're very underpowered, so I don't think it's blood type.
So some people produce less sebum than others, dry skin versus oily skin. Does the sebum level affect the degree of attraction? And if so, does detergent treatment of skin, deodorant, soap versus lipid-sparing soap make any difference? Mm-hmm.
So we didn't quantify that parameter. Every person vigorously washed their skin with unscented soap before they put the stocking on and wore it for 6 hours. But again, everything was I mean, skin type does vary, and microbiome varies across the body, but everything is the volar forearm, which, I mean, in my limited experience, it's I think, less subject to those kind of variables of dryness, maybe, but yeah.
Well, we have more questions that we can possibly take because it's already time. So I would like to, on behalf of everyone, thank you so much for this wonderful, wonderful talk. I'm pretty sure I'm like subject K. That was what I was telling myself.
Congratulations.
Now I don't know. But thank you so much for coming and really giving us this wonderful information. Thank you so much, Dr. Vosshall.
Thank you very
Thank you.
much.